microevolution. But before we talk about microevolution, we need to talk a little bit about macroevolution, because that's what microevolution is not, right? So macroevolution. That's what you guys are probably familiar with when we talk about evolution. Macroevolution, macro means large, right? So these are the large-scale changes between species and among species they take a really long time to happen. They happen in a very long period of time, and they can result in new species being formed. Okay, so these are large-scale changes that happen over long periods of time. And may result in speciation. Speciation means the formation of a new species. Okay? That's not what we're going to talk about today, though. What we're going to talk about today is how macroevolution actually happens, which is a whole bunch of microevolution over a very long period of time. Microevolution is much easier for us to observe. These are the changes in allele frequency from one generation to the next generation. And I want to talk about this this term here, allele frequency, for a second. When I say allele frequency, I'm not talking about the frequency of any particular phenotype, right? So let's say, theoretically speaking, it's not true, but let's say that everybody in this room had brown hair, okay? That doesn't mean that the frequency of blonde, the frequency of the blonde allele is 0 in here. It means that everybody has the phenotype of brown hair, but there could still be blonde hair alleles hiding out in people who are carrying that, people who are heterozygous for brown hair, right? And so changes in allele frequency are going to be a lot slower than changes in the phenotypes, because you can also always have those recessive genes sort of hiding out with the dominant phenotype. Okay, so there's lots of things that can do this. Sometimes these changes in allele frequencies are the result of a directional change, something that's actually like forcing it towards one phenotype or the other. Okay, so may be a directed change. Okay, so one type of directed change is natural selection. What's happening in natural selection? What is actually doing the directed change? What is forcing it? Yeah.
Is it death?
Death caused by what?
By the environment.
By the environment. So we call that environmental pressures. So environmental pressures for natural selection are going to favor one allele over another. Okay. There's also artificial selection. Artificial selection, what's that about? Yeah, like who's doing the directing there? What is doing the directing?
Other organisms?
Specifically, what other organism?
Humans.
Humans, yeah. Okay, so in this case, this is humans. favor one allele over another? So I think the question was asked in here yesterday as to how long does this take? How long does the process of evolution take? And the key to that is it depends on the mortality rate. So if one allele is being selected for by an environment, right, for natural selection, but the mortality rate's like 5%, then that's going to take a really long time to clear out the other allele. But if we're talking about artificial selection, a lot of times when humans do things, we go full out on it. So the mortality rate's like 100%. If you consider like consider dogs, okay? Dogs are the common ancestor of dog is the wolf, right? And the way that we got dogs are very recent as far as like on the geologic time scale. And the way that we got dogs was these hunter-gatherers, these humans, they were like cooking their food on a fire, right? And they'd cook their meal. And wolves are very afraid of strange-looking creatures like humans, especially ones that can make big, bright fires. Okay, so for the most part, the wolves would stay away because they had high levels of this stress hormone that sort of makes them anxious and causes them to be aggressive towards anything that they're afraid of. Okay, so some of them naturally had a lower level of that stress hormone, so they were more likely to come up to the fire, come up to the people who were eating, and you know, the humans thought that was funny and cool, so they would like throw them a bone or something like that. Like, I'm not going to eat this anyways. Have this bone, and the wolves were like, "Ooh, cool! This is free food. I like this, right?" And so they continued to hang around. And the ones that had the least amount of that stress hormone might have just stayed there. They might have just hung around for good, and the humans kept feeding them. Right? And if they did, then the humans might breed them, might allow those wolves that were staying around to breed together, and they might have offspring. And so some of those offspring might have the predisposition to like come up and get close and cuddle, and some of them might have the predisposition to like bite you, right? And so if they ever bit them, they just killed them. They're like, okay, throw that one in the fire, right? That's not a good puppy. And then the ones that didn't bite, the ones that were more cuddly, they just kept those around and they bred them. And so this was a 100% mortality rate for aggression because the humans would just kill them, right? And so it doesn't take very long for those genes to get filtered out of that population then. If anytime one of the members of that population is aggressive, you just kill it, right? So it's very quick, gets genes out of the population very quickly. It's worth noting, however, that natural selection and artificial selection often have very different interests, right? So if you take, you know, cute and cuddly, you know, Jean Frise or any of the little like toy dogs that we have, and you like put them out in the woods that also has wolves, it's not going to go well for them, right? The dogs that are cute and cuddly and small, they have been bred to be our little, you know, domesticated toys, and the wolves are still wild animals that are surviving on their own, and so they're, you know, have aggression and strength bred into their bloodlines and not short hair and cuddliness, right? So different things we're looking at here. Humans are selecting for things that are not necessarily good for survival, but are good for living in our homes and having our dogs not eat us. All right. So other things for this, you may also have non-directional changes, random changes. So the first example of this, the most prominent example of this, is something that's called genetic drift. And what genetic drift means, and you're going to hear me make this analogy like 1,000 times this unit, sometimes when you flip a coin 10 times, you would expect, first of all, you'd expect flipping a coin 10 times you should get 5 heads and 5 tails because there's a 50% chance to get either side, right? But sometimes when you flip a coin 10 times, you get 10 heads, right? Sometimes when you flip a coin 10 times, you get 10 tails, right? And that's, you're not breaking any laws of physics, right? That's just like something that happens due to random chance. There's a small probability that you can get 10 heads in a row or 10 tails in a row, okay? This also applies to genetics. So we said that in a monohybrid cross, right, you have a heterozygote parent and a heterozygote parent, that you're likely to get one offspring, 25% of the offspring will be homozygous dominant, 50% of the offspring will be heterozygous, and 25% will be homozygous recessive, right? It's possible, though, that if two heterozygous parents have 10 offspring, that 100% of them will be homozygous recessive, right? It's not highly probable, but it's possible, right? And so that's what genetic drift is all about. Sometimes just random chance causes gene frequencies to shift, right? Sometimes when you flip a coin 10 times, you get 10 heads, right? So changes in allele frequency. Due to random chance. Okay. Now, thankfully, there's this thing called the law of large numbers, and when it applies to probability, and that says that if your sample size gets larger and larger and larger, you should approach the actual probabilities. So, for instance, if I flip the coin 2 times, it's pretty likely that I'd either get 2 heads or 2 tails. But if I flip the coin 10 times, it gets less likely. 100 times, it gets even more less likely. If I flip it 1 billion times, I should end up with approximately 500 million heads and 500 million tails, right? That they equal out if you have larger sample sizes, right? Okay. The other one's called gene flow, right? And gene flow has to do with the mixing of populations. So when populations mix, the allele frequencies in both gene pools will change. Okay? So when I think of this, I think of like actual pool, right? So gene pools, right? Gene pools are all of the alleles in a population. But if you think about actual pools, like let's say somebody has like a saltwater pool, right? One of those new fancy saltwater pools, and somebody has like a normal chlorine pool, right? And then there's a big rain or something like that, and the pools overflow, and they like literally flow into one another, right? That means that the freshwater pool is going to be slightly salty now, and the saltwater pool is going to be slightly more freshwater because the populations have mixed, and that is not the result of any type of selection. It's just like the populations mixed, so the allele frequencies changed, but it doesn't mean that there's any directional change. It didn't happen for a reason. They just mixed, right? Okay. So these concepts apply to something that's called Hardy-Weinberg Equilibrium. Hardy-Weinberg Equilibrium says that there is this condition, this state that an allele might be in, or that a gene might be in, where the allele frequencies stay constant. Okay? So allele frequencies for a particular gene will remain the same as long as and then all these conditions. Okay, so number 1, populations, or the population is large or infinite. Okay, so having a large population, which one of these things up here is controlled by having a large population? Yeah. Nope. Which one of these possible changes? Genetic drift, yes. So we said that if I flip a coin 1 billion times, I should get 500 million heads and 500 million tails. So the larger the population, the less likely that I'm going to have a change in the allele frequency due to random chance. Okay, so this controls for genetic drift. Number 2, okay, there has to be no selection. Meaning the trait does not affect survivability. Trait has no effect on survivability. Okay, this obviously controls natural selection mostly, because that's the type that's going to control survivability for the most part. So it controls for natural selection. Condition number 3, random mating in regards to that specific trait. Okay, so this has to be a trait that is not only not selected for in sexual selection, but also can't be linked to anything that is sexually selected for. Controls for sexual selection. Number 4 says no new mutations. Okay, because if a new mutation pops up, then that's going to add another allele to the gene pool, which is obviously going to change the frequency because now you have another allele to consider, right? So this controls for new alleles, which should generally be the result of SNPs, single nucleotide polymorphisms. And then number 5, no mixing of populations. And that obviously controls for gene flow. Okay, and if all these things are true, then you can have a population that's in Hardy-Weinberg equilibrium. So how likely do you think that all these things are going to be true? Not likely at all. So this very rarely happens, like almost never. Okay, but there are some instances that it does happen, right? So interestingly enough, one of those things, what's up?
When you say controls for gene flow or like controls for whatever the word is, what does that mean?
It means that it's not going to allow that to affect the population. So like when I say controls for genetic drift, the fact that the population is infinite means that genetic drift is going to have no bearing on that allele frequency. It's not that genetic drift is not likely to happen. Okay, so there is actually a couple of alleles for the human population that are, for the most part, in equilibrium. And so we're going to do an experiment right now with one of those. Okay, so in this class we're going to do an experiment, and our experiment is going to be the frequency of attached versus detached earlobes. Okay, so first of all, a couple things for this. We can say no mixing of populations because we're just kind of going to assume that the population is global, that the human population is global, right? There's no new mutations. There's no mutation that I know where you just don't have earlobes. So you have them attached or you have them detached, right? And then no selection. Your survivability is not dependent on whether or not you have attached or detached earlobes. And then random mating in regards to that. On Match.com, there's no drop down for like earlobe preference, and you don't like go to your boyfriend or girlfriend and be like, ooh, detached earlobes, yuck. And then yeah, so it really doesn't matter. So I want you to take a second, and I want you to figure out if you have attached or detached earlobes. Attached earlobes mean you don't have any like dingly danglies right here, right? If you do this, it doesn't do anything. Okay, you might need to rely on your table partner if you can't figure it out. If they are detached at all, I want you to consider them detached. Okay, so raise your hand if your earlobes are detached. Woo! This is going to be messed up. All right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. Oh, man. All right. Detached earlobes, 23. Attached. Raise your hand. 1, 2, 3, 4, 5. Ooh. Okay, so ours is gonna be a little messed up in here, but that's fine. All right, it's gonna work. So detached earlobes are the dominant trait, obviously. So you can figure that out. Okay, so that means that the phenotypes are big E, or big E, little e. Okay, are they attached? All right, so we'll be 24 and 6. That might help a little bit, but probably not a lot. All right.
Wait, what did you 22?
Oh, sorry. Yeah, 22 and 6. My bad. All right. So that means the people who are of attached earlobes, they are homozygous recessive. So they're going to be only little e, little e. Thank you so much. So these are little e only. Okay, so now we're going to go through and figure out our frequencies for it. All right, so the frequencies, we're talking about phenotypic frequencies here. Okay, so we've got 22 divided by a total of 28 people in the class. So that gives us 79.79. And that tells us that this is going to be 0.21. Okay? All right. So then, we want to figure out the frequency of the alleles. Okay? In order to calculate allele frequency, we need to use a formula. And the formula looks like this. Hardy-Weinberg Formula. It is p squared plus 2pq plus q squared is equal to 1. Okay? And in this, p squared is equal to the homozygous dominant genotype. Q squared, or sorry, 2pq is equal to the heterozygous genotype. And then Q squared is equal to the homozygous recessive genotype. Okay, and I'm going to show you really quick where these come from, because I think it'll make a little more sense and help you remember it a little bit. So if you have a Punnett square like this, okay, and you have a monohybrid cross like this, you end up with something like that, right? So in this first box, you have A times A. What's A times A? What's that? It's A squared, right? So if we consider big A here to be P, that tells us that that's P squared, right? So that's the homozygous dominance. Then you've got big A times little a and big A times little a, which is 2 big A little a, which corresponds to 2 PQ, because P represents the big A and Q represents the little a, right? And then down here, you've got little a times little a, which is little a squared, and that goes in here to Q squared, right? And we know that this is going to add up to 100%, and 100% is 1, right? We see where this comes from? All right, there's one other thing that we can assume, and that is, in order for this to work, there can only be 2 alleles that we're looking at. There can't be a third allele, so we can't have multiple alleles, right? So that tells us that if you have P plus Q, that should account for all the alleles, so P plus Q should be equal to 1. Yeah?
Do we have to memorize that formula? Or
I believe it's given to you, yeah, it's given to you. All right, so, and you definitely need to know how to use it, though. So let's go through and figure out how to use this. So in this equation here, we know just Q squared, right? That's the only number we know, because Q squared is the homozygous recessive genotype, and we know that the homozygous recessive genotype was 0.21, but the 0.79 that we have is actually the combination of the homozygous dominant and the heterozygote. So we only know this, right? So we know Q squared is equal to 0.21. So if I know that Q squared is equal to 0.21, can I find Q? How do I find Q? So the square root of Q squared is obviously Q, and so that tells us that's the square root of 0.21, and that tells us that Q is equal to the square root of 0.21, which is 0.46. Okay, so then I know that P can be found by taking 1 minus Q. So P is equal to 1 minus 0.46. So P is equal to 0.54 then. So I know that the frequency of the big E allele is 0.54, and the frequency of the little e allele is 0.46. So if I asked you the question then, estimate the number of people in this class that are heterozygous for this trait, what would you do? Yep, so you go 2 times 0.54 times 0.46. And we get, like, basically half, right? So that tells us that half of the class, which how many people were in the class? 28. So that tells us that 14 people in here are, 14 people in here are going to be heterozygous. Yeah? The 2PQ, the one that we just did? The heterozygous thing?
No, yeah. How you got like the 2 and then why you like multiplying?
This thing? Okay, so for the heterozygotes are represented by the 2pq, right? So I took 2 times p times q, right? 2 times 0.54 times 0.46, and that tells us that roughly 50% of people in the class are heterozygous, right? And then, so that was 14, I said, right? And then if I take 0.54 squared, oops, that's none of those things. 0.54 squared, nope, squared.
There's 2 decimals.
We're okay. That tells us that like 30%, so that would be 0.3 times 28. So like 8 people, roughly, would be homozygous dominant, and that tells us that 6 people should be homozygous recessive in here. Which it was actually 7. No, it was 6. So that's perfect.
Is that like for sure, or is that just...
No, it's an estimate.
Oh. Okay.
It's an estimate, yeah. So, if I go back and look, though, like look at last period, we ended up with actually pretty much the opposite, right? We ended up with 0.54 as little e, right? Not as big e, right? And then in the period before that, we ended up with 0.56 as little e, right? So why does that happen? Why is it different? What condition did we not meet?
In this class
Yeah.
specifically?
Yeah.
We had less people with the attached earlobes.
Sure, but what Hardy-Weinberg condition did we not meet? Yeah, the first one. The population in here is not large or infinite, right? It's 28, which is very small. N equals 28 is virtually nothing. It's possible that later on, at some point in my teaching career, I will end up with a class that has 100% detached earlobes or 100% attached earlobes due to random chance, right? So whenever they assign your schedules, for some reason in that year, it will assign me 30 people that have attached earlobes. And that'll be super weird, but it'll be cool, right?
You should ask
So
I don't think that's on your like permanent file or anything.
like when you sign up for Inkbot, you should just be like, oh, you have detached earlobes.
Oh, BT dubs.
Yeah, so
Attached
check
to
a
detached
box.
earlobes.
What are your earlobes?
Neither.
I feel like I would appear on the news or something if I started to ask that. All right. Questions on this? All right, that's it.